Hello, welcome back to Brains, Minds, and Consciousness. Today we're going to start our little walk up the mountain of brain and mind and consciousness. Chosen to use this metaphor for our hike through this course content. So, on the pyramid, the climbing height of this mountain, we start with brain. We will work our way up to mind, and we will finally sit at the top and ponder some things about consciousness. So, to start, we are here. And to really start, I think it's best to go way back to some of the first questions that were asked in a methodical way about brains. So, let's begin. Why the heck should we care about brains? You know, it's a pretty old question. I think we all have a better sense now why you would want to care about what a brain do. We nowadays would say things like, because it's the physical basis for the control of behavior and the mind itself, dang it. But that's not always been clear. Even the basic understanding that brains affect behavior directly was not always obvious. We didn't always have the ability to measure what these things did. I think we all knew that if someone's brain came out somehow, it wasn't good for them to survive. So it's always been known that it's a sort of an important organ, but it wasn't clear how and what and why your brain affected your behavior. One of the most interesting historical cases that begins this discussion in sort of a more medical sense and thus a little bit more scientific sense is the case of our old friend Phineas Gage. You may have heard of him. It is a very famous scenario. So it's one of the earliest and most cited pieces of evidence for the idea that if your brain is affected by something physical, that is changed physically, then your mind might change along with it. So here's what happened. Phineas Gage worked on a railroad construction crew. He was the foreman. And he suffered an accident with a tamping rod. Tamping rods are sort of these long metal poles that you use to indirectly prod at and pat down various, dirt, and say you're burying explosives, for example. So an unplanned explosion launched the rod right through his face and out the top of his head. I know it's grisly to think of. This is a reconstruction of what we think this injury would have looked like. Brain exploding skeletons. That's what you get in this class. So, yeah, it was a grisly thing. So, the thing is, though, he survived. And he recovered normal motor function in a few months. So that is evidence straight away of, okay, the part of his brain that was damaged allowed him to, he still has all of his motor functions. He did have to relearn them, but he got back up to speed. So the trouble, though, is that this particular part of his brain may have been in charge of certain other things about him. So popular accounts, now this is the trouble, that it's hard to tell exactly what was going on back then. We didn't have, people weren't looking for this. It was only in sort of retrospectives that people noticed what was happening with this guy. But they said his personality was completely changed. The way he interacted with the world was very different. He was less inhibited. He was much more aggressive. People said he was close to base animal instincts. But people were a little more prim and proper in certain circles back then. So maybe he was just a little more ragged. So the accounts vary. It's not perfectly clear what everyone in his life thought of him. But, and again, it's also not clear because we didn't have any good imaging of him back then, but it's unclear exactly what the state of the damage was. But the idea is he changed. This pole went through his brain. He survived, and he became a bit of a different person. There's another example. Henry Molaison, HM is what he's known as. This is another brain affecting mind scenario. In 1953, he had a radical surgery to relieve epileptic seizures. If you don't know what a seizure is, what an epileptic seizure is, it's when a lot of neural activity happening in the brain spreads kind of uncontrolled. Normally your brain has lots of mechanisms for tamping down on one neuron firing into the other, but in a seizure, it's sort of this cascade of, oh crap, we can't stop this. And so having a seizure is a pretty serious thing. It often comes with motor spasms, and it comes with lots of strange effects on memory and on your attention and everything about it. So they're a pretty bad thing. So he had this surgery to try and stop it, right? And I think all they really knew back then was there's a lot happening in there. Maybe we could remove certain parts to hope it to slow it down and stop it. So removed a large part of his brain. Some really important pieces that you may have heard of before: the hippocampus, the amygdala, and other nearby areas. The great thing is that the seizures actually did stop. The bad thing was that he now had what's known as severe anterograde amnesia, as in he was unable to form new memories. And to give a bit of an illustration here, above you see, look at these two, the blue and red bars. The undamaged memory is just, you proceed as normal. The anterograde damaged memory, you have, at the time of the damaging event, the surgery, in HM's case, everything before the surgery is intact. He still remembers all that. He still remembers everything about the weeks and months leading up to it and his life before that. But from then on, he could only form memories that lasted about 30 seconds, even into his later parts of his life. So he had this full intact memory, but because some pretty important elements of his brain were removed, he couldn't form new ones. That's a lot. If you've seen Finding Dory or Finding Nemo, or you've watched Memento, these are examples of that. Although Dory has some memory of what's happening, but it's an example of anterograde amnesia. It's hard to form new memories. This had a lot of consequences for HM. He didn't recognize himself in the mirror, especially as he started to age, right? He's graying and his beard's changed and his hair, I'm sure, his face. And it was tough to interact with the world too, because he couldn't remember people who left the room and returned just a few minutes later. He also had some spatial and motor impairment, but it's not clear how that connects directly to those specific brain regions. One thing to know about the brain is that the regions, though they do different things, they're all kind of wound up together. So damage in one area might affect other areas in different ways. But an interesting thing was he could still learn certain things. He could still learn motor skills. So he might not be able to remember his painting lessons, but he would retain the skills he acquired during those lessons. Might not necessarily remember Bob Ross, but he would still have skill. That must mean there's different ways to acquire memory, because skill is also, of course, some kind of stored knowledge, some kind of stored pattern of activation that you can reuse later, some kind of synergy to help you make sense of how the pencil hits the paper. Also, I don't know if he took specifically drawing or painting or anything, but the idea was that these are the kinds of skills that he could still acquire, even though he only had a memory of 30 seconds at a time. So, getting mind out of brain, Phineas Gage and HM represent these two sort of medical wonders. They were fascinating at the time that they occurred because they were somewhat unheard of. You know, these were experimental surgeries or freak accidents that normally you wouldn't survive. And it's this shows that it's not just that the mind and brain are related, but how they are related. That there are specific ways that a brain can be damaged and the ways that can affect the mental experience. So it doesn't just kill you. You know, we might have once thought that, right? Hit person on head with rock, no more person. But more, it changes how your mind works when your brain becomes damaged. So that's a major theme here, to understand that there are functions of the brain and the way that the pieces all fit together that, you know, your mind is composed of, right? We can also talk about more of the biology, so we can really drill down. If we want to understand how a brain works, and we really want to get down to the nitty-gritty details of how all the mechanisms move and how the moving parts exist and came to be, we have to first invent the universe. So, right at the start, let's do a brief history of life on Earth. All right. Here is a bar that represents the timeline of Earth's existence from today. So today's at the bottom in the red. The top is the gray with the very beginnings of this rock. Let's go. So, about 4.6 billion years ago, Earth formed. 4.1 billion, the atmosphere formed. Oh, that's kind of nice. That's useful for us. And then somehow in that soup of the water and the atmosphere and the roiling mass of this Earth, 3.9 billion years ago, we got prokaryotic life. That's important because that's when bacteria and other archaea, as they're known, started to form. Bacteria are the first organisms that are cellular. They are collections of pieces, right? From there, photosynthesis starts to come around, you know, trees, and well, not trees to start, of course, but cells that are able to take sunlight and use it for useful things. So these chemical reactions that turn light into chemical energy to be used and stored. I promise this all is critical. Now, but that said, you don't need to you don't need to memorize every last detail here. You know, this is not the focus of the course, but the details help you chart the trajectory. So, continuing. We get photosynthesis at 3.5 billion years. 3 billion, we get oxygenation. Well, that's where we can start changing CO2 into oxygen, and that's as a byproduct of photosynthesis, making it so that the planet could be coated with oxygen and, starting to poison all the anaerobic organisms. So that means something else must come up in its place. This is where the eukaryotic life begins. And these are the cells that have a centralized something, a nucleus, a bundle of the important information stored centrally so that it can do things like replicate and other important details about how to run the cell. Okay, so now we take a long time, about 1 billion years, before these cells start to glom together. Multicellular life. Organisms composed of lots of cells, and they all are starting to do different functions. Aha! Here we're starting to see, oh, there's something important about how this maps the brains here. And from there, finally, as these cells start to sort of distinguish themselves and make sense of being different little workers in this mechanistic pile. We finally acquire vertebrates, which is where the spinal cord develops to protect certain types of cells and to sort of extend the central blob of these interconnected cells into the world. The spinal cord is the architecture on which your things like muscles and obviously the bones that come with it, of course, but they all evolve together. The idea that you can extend into space. So from vertebrates we get mammals, from mammals we get genus Homo, and from genus Homo we get modern humans. They are us hanging out these days. So, woof. This is the history of the Earth. This is life as it has processed forward and changed, and that's where we finally are, right? We exist. We have spinal cords. We are multicellular. We have centralized information stored in things like DNA. Okay, so let's hit some of these highlights. In the beginning, everything was about energy efficiency. Collaboration is usually easier. The more things that kind of glom together, the more we can enter low energy, stable states, right? So atoms were just free-floating bits of stuff. Hey, that's how Earth was to start. Cool. Collection of rock and made of atoms. Atoms like to combine into molecules. This is, basic chemistry. And basic physics into chemistry, right? They want to be connected. There's something about that energy state that draws them together. It's easier for them to exist in the world when they are connected. Molecules, by the same properties, would then start to form into protein chains. I guess it's easier to float around in the goop and bump into more molecules if you are in this chain form. Proteins then started to develop cell walls so that they could have maybe a little bit more protection. They didn't have to get eaten and absorbed by everything else, right? So having a wall makes them into prokaryotes, the first sort of rumblings of what you could think of as a specific entity. Now here's a trick that was really important: chemotaxis. That is what allows one of these systems to detect good and bad chemicals. So the things that might erode and destroy something caustic to these proteins and their cell walls, and things that might be good for them, things that you maybe absorb more material, right? Beginning to seem like making a decision in the world, like putting some kind of decision tree on where this thing ought to float. But that only matters if you have mobility. So allows these little organisms to start to chase prey and avoid predators in the same way that you could detect good and bad chemicals. Aha! Now we have input and output. We have the ability to detect things in the world and the ability to affect the world, moving, expending some kind of energy that is absorbed into this cellular system and doing things with it. Fantastic. So now eukaryotes evolve, which gives them the nucleus, and that allows them to protect their genetic information. How do I be the thing that I am? DNA, right? And that leads to sexual reproduction, allows for the exchange of DNA, leading to, this is the key, this is where rapid evolution can begin, because there's a lot more kinds of information being exchanged between the cells. They are able to give the information to each other and make something new that will try a different way. That's the explosion of variation over the generations, as opposed to just everything sort of always being the same copy and just trying to make it. Now we have the ability to alter and change and morph and move and find niches. And that's the evolution of life. All right? Cool. So we've invented life. Fantastic. We've seen its trajectory from rock into something that interacts with the world and can change. Okay, specialization is afoot. So that's how we've moved through the different stages of life coming to be. Now the question is, where do neurons fit into all this? They seem kind of funky, right? They seem like there's a lot of, they're very specialized, and they are responsible for a lot of really complicated things that, I don't know, a bunch of proteins floating around in the goop don't necessarily need, right? Well, the idea that you might want to have cells that can communicate to one another is the key. And it turns out it's pretty dang useful to have that. So let's talk about the underpinnings of neurons. So we talk about chemotaxis, right? The idea that receptors can be sensitive to specific chemicals, right? I'm just trying to avoid, hey, I don't want to walk into that acid or perhaps that extremely basic solution that will more or less dissolve me. Let's not do that. That's what chemotaxis was good for. So intercellular communication, though, the idea that cells can actually communicate with each other using this technique and not just avoid the acids of the world, but talk, send a message of some kind. Just the basic idea of transmitting information. That's where we have sort of bootstrapped chemotaxis and pulled ourselves up into, oops, now we can talk to each other. So the idea for multicellular organisms, you can release hormones. Now these are specific combinations of small bits of matter that perhaps communicate specific different things. For example, in you have adrenaline, serotonin, dopamine. These are all hormones that your system can release to send a big message to the rest of your body. Hey, something's going on. Perhaps it's time to sleep. Ooh, this was good. Everyone feel good. Or perhaps adrenaline. Oh crap, there's a tiger. We have to go. Move those muscles now. I don't care if they're ready or not. Right? So hormones are the basic mechanism of sending and receiving between cells. So you would send these chemicals, and the other cells have to have the receptors to receive it. Now, this enables the concept of cell-to-cell communication, right? And how do we do that? How do we manage it? The easiest method is just to plop some hormones out into the world, and all the things nearby will notice. That's sort of the basic chemotaxis version of this. So, beyond, again, just detecting good and bad, you can just get information. But the idea is that more hormones are going to reach the closer cells. There's not anything useful here about moving long distance. You can't really make long distance work here. You'd be lucky if a cell that's 10 times further away actually receives your message. So that's not enough. Just having things spread out in very, very proximal ways leads to everything just being kind of a blob of soup. It's hard to have any kind of structure, right? If we want to be able to have a spinal cord that can actuate big old meaty legs, then we need the ability to target specific cells, talk to specific cells at a distance. So we develop these long protrusions, okay? We may have heard of the cell body and then the axons, the parts that stretch out from the neuron. And the idea here is that this allows for very fast and targeted communication. Okay, so now we've kind of hacked the system to say, hey, I can reach people right next to me and I can reach people far away from me. I say people. I'm anthropomorphizing these cells. You shouldn't do that. They are just little goopy boys. But that's still, this is it. This is the idea. And to get into the exact reasons of exactly how these things evolved, it's a little tricky It's a discussion for another day. But this is the idea. This is how I say as multicellular organisms. So, we can be in it with dogs and dinosaurs and all that. This is how we gained the ability to do this really cool thing. And this is the beginning of the neuron. So, now we have to start asking the question, again, in broad strokes. This is just our first touch of kind of circling the mountain, getting the picture of what we're walking up. How do these things fit together in the brain? How do neurons fit together? What's the exact sort of, perhaps not exact, but the arrangement of them? So, there are two types of brain cells: neurons and glia. Let's define these. Let's look into them. Neurons. These are the cells that you've heard of, our old friends. These are the ones that can receive, process, and transmit information. They have a cell body, just like every cell that came before it, and they have two types of branching extensions. Dendrites are sort of what you think of as the input to the cell. These are short, thick, many branches, thinning as they get further away. These are the areas where the receptors live. You want branching so you can have a lot of them, a lot of receptors ready to receive information from anywhere. The axons, on the other hand, are the output of the cell, the long, skinny boy that runs off to somewhere else so that it can then transmit something. One long, thin extension that, again, terminates in a complex branch structure so that, again, you want to be able to send your little chemicals, your little hormones, multiple different areas. You might want to hit several other overlapping cells. This is a good image of a neuron. This is kind of how they look. There's a lot going on. They look kind of like trees, right? That's that organic fractal nature of the things. So these are the ones that we've all sort of internalized. We kind of get in a basic way how neurons work. We'll cover it in more detail, but you can understand that they take information, a neuron fires somewhere, and then something happens, and then the neuron will fire off somewhere else, and information moves. Cool, great, useful. Glia, on the other hand, are the cells that exist to support and maintain and facilitate the function of the neurons around it. So there's lots of varieties with different functions. Some are just there to hold neurons in place so that they don't kind of wobble around and flow. Oh, hey, I'm from the motor area. Let me go touch your memories. That's not necessarily good for the neural processing, right? So you need something to help with the structure. Sometimes you need them to connect neurons to blood supplies, as in these neurons are still cells. They need to have some kind of energy to burn to be able to do their job. So they need something to come up to connect them to the baseline, the heartbeat down there somewhere near the stem. So there's also the fatty types of glia that create myelin sheaths. And we'll talk about how those work later, but it's the simple idea that you don't want much leaking out of the cell, out of the axon. In fact, it makes it a little faster if you can sort of insulate it. So that's what the myelin sheaths do. They help kind of increase speed and efficiency of a neuron's axon in processing and sending its signal on. And regulating blood flow, just in the same way of, having a little bit of a more infrastructure to make sure all these happy neurons that do all this cool information work are moving and powered and so on. Another structure that we should talk about is the synapses. These are the connections between neurons. So, as we said, we saw that the axon spirals off into a whole bunch of terminals, the little bulbs in that little network, and they hopefully are overlapping and close to the dendrites of other neurons. So, the the connections between them, between neurons, sometimes they are in direct physical contact. But regardless of the fact that they are touching, they are still always chemical in function. So the way one neuron activates the next neuron is by sending a hormone out into that neuron. So the axon terminals get the impulse from the cell body that say, oh, wait, we're firing. And then they will emit specific chemicals that the next dendrites of the next neuron will catch because they are sensitive to those chemicals. Remember, chemotaxis. It still all boils down to that. Receive them and then carry the message forward by saying, ooh, we're getting enough signal from other cells. Let's fire ourselves off. So, the current take in neuroscience is that most brain function is in terms of synaptic function, right? It's sort of a dicey question to begin with, but it's the idea that synapses are where the action happens, the transmission, the information. If it weren't for a connection between the cells, then whatever happened inside a single neuron wouldn't matter. So this is where all the important stuff happens. A lot of mental disorders are explained in terms of synaptic dysfunction specifically. So that's why most drugs will affect synaptic function in some way. So things like SSRIs, right? You may have heard of these. This is a common treatment technique. And we'll get into more hormonal treatment mechanisms later. But the idea here is that SSRIs are selective serotonin reuptake inhibitors. Let's break each one of those out to make sense of it. Selective means that only certain cells will be sensitive to this chemical, the SSRI itself. Meaning it's trying to target certain areas of the brain. How? Leave that out the side. Serotonin is the hormone involved, and it's the one considered to be responsible for things like depression. The idea is that you have too little serotonin active in your system. This is sort of the feel-good hormone. And a brief aside on that, serotonin is not strictly, quote, feel-good, right? It does all kinds of functions in the brain, but it's considered to be part of this process of the whatever is causing the spiral of depression. So serotonin is the hormone involved. Reuptake is the process of one neuron, the neuron that fired the hormone, taking it back, saying, oh, you're done with that? Okay, I need it back. I'm going to break it down for base parts. And then the inhibitor is that this chemical, the SSRI itself, will dampen that process. So to recast this, SSRIs will prevent certain neurons from recycling too much serotonin, meaning more of that chemical will be present to help activate nearby cells and promote whatever good thing the serotonin has been doing, and perhaps what has been lacking in the depression patient. So that's just an example of how we as a species attempt to use our knowledge of the ways that neurons interconnect and talk to each other for good, right? Trying to help people out who are in a major way. So, yeah. Moving on to some distinctions that we should probably note, or perhaps lack thereof. What is the actual difference between neurons and other cells? When it comes down to it, structurally, they're almost the same. They have all the same parts. They have cell walls. They have mitochondria, the powerhouse of the cell, right? Hormonal sites to create different hormones. They just have much longer protrusions. So whereas a normal cell wouldn't waste all that effort trying to stick out and be all wiggly, it just kind of balls itself up into a little globule. The neurons go out of their way to be longer for reasons we already discussed. But there's some other things that you might want to know about neurons. For one thing, they're hungry. Some people have called them the hummingbird of cells. So they are 2% of your body weight So if I split you up into your constituent parts, the pile of neurons would be 2% of what you weigh. But they consume 20% of your oxygen and 25% of all your glucose, your food, your energy. Whoa, they are hungry. They are doing a lot of work. So unlike other cells, they'll die very quickly if they're deprived of oxygen. That's why holding your breath too long is a bad idea, and sometimes you die. Because your nerves say, oh, dude, we need that so much, please, oh, God. Whereas the rest of your cells can hang out. They don't necessarily need that oxygen so much. But these ones, they need it all the time. They also aren't especially good at regeneration. They can, some people have told the story, oh, that neurons never regenerate. Well, certain ones of them can. A friend of mine got hit by a boat. A motorboat ran over him in the waters, and he lost feeling in his thigh for a long time, but it came back. So, hey, that's just an anecdote, but it goes to show that neurons can regenerate, but they're not good at it. So, a rule of thumb, pardon, is that at 1 year old, you more or less have all the neurons that you're going to have. Loss of these neurons is totally natural. And they're going to grow and change, but sometimes you start to lose them with age, right? Might be fewer branches between neurons. They might shrink in size. The overall volume will decrease, which, we know, interestingly, because we're living longer than we used to. Ancient times, it was unlikely that you'd live to up to 80, but nowadays, most people tend to live into their 60s. So we're able to see more of how these different processes happen. The process of these neurons kind of winnowing down with age. So, brains. We've seen how neurons came to be from sort of the beginning of all life. And we've seen how some folks suffer when their neurons are damaged or removed. But another way that we can couch this question is by looking at a modern disease that's continuously a problem, and that is Alzheimer's. So, Alzheimer's disease, it's a rapid increased loss of neurons. Some loss is natural, but Alzheimer's will accelerate it. Some of you may have family members who have had this disease. I had one myself, my grandmother. It's kind of a terrible thing, but it helps us to see. And it maybe helps to understand how it works and what's happening. So the way it begins to present itself, and this is the sort of classic one, is that it usually starts with memory deficits, right? It's harder for them to remember things. They might start confusing things they saw on TV with things that they did in real life, that kind of stuff. But what follows is broader decline in cognitive capabilities. The ability to actually think and process information starts to go away. And then finally, a rapid loss of motor function and speech. And perhaps more critical, necessary life functions like gastrointestinal things. So it's a long, slow decline typically, and it is a sort of a curious disease. It has for a long time we've understood dementia in the broad sense, but Alzheimer's is the specific flavor of that. And it is this idea that your neurons simply cease to function. And it's not clear exactly what happens. There's competing hypotheses about the cause, and so it's hard to prevent it, right? Because it's a very different thing If this is just the normal decline process that you see in neurons because they have a sort of half-life that is accelerated by some other process, or if it's a specific disease that is directly affecting neurons and damaging them. Or perhaps it's some combination of both. Maybe you have the disease that triggers the decline process, right? And on top of that, it's difficult to study the disease because it plays out over this very long, extended time. You only really notice that it's starting to progress and happen to you or your family members when it's obvious, right? When it's actually already having effects on your life, on your mind, on the way that you interact with the world. It's like, oh, grandma's getting a little, it's getting harder for her to remember things. Should we be concerned? It makes it very difficult to study because we usually miss the beginning of the disease, the onset. What are the conditions under which it starts, because it's very slow. And the progression of it is very slow. It's this expensive process. It's difficult to maintain their life, by treating them, but it's also difficult because we're constantly trying to treat them to understand the thing that we're treating. It's this sort of catch-22. So this is a good example of why it is so important for us to understand how the brain works. This is open research. This is still happening. Alzheimer's disease is still at the top of the list of diseases we're trying to tackle. So I think it's important to recognize that this is a brain disease, and understanding how and why the brain does what it does is a great step towards understanding these particular kinds of diseases. So, this has been an introduction to brains. We've seen how public consciousness began to form around these interesting questions about brains and minds, starting with Phineas Gage and up through H.M. A very fascinating scenario. We looked at the development of life, neurons, and everything. That's been nice, I hope. A tour of the history of the world. And we examined a more modern struggle to understand this degenerate neurological disease that affects millions of people, Alzheimer's. So, I hope that sort of gives a broad introduction to brains and why we think they're kind of neat and important. And as is my way, I'll leave you with a final thought. Consider the two abilities that cells acquired through evolution: the ability to detect things in the world and the ability to move in the world. How does this relate to the basic properties of neurons? How does the transmission of information fit into this picture? All right, that's it. Thanks, and take care.